about immunity and the plant immunity and the endoplasmic reticulum. So, okay, Emily, let me just switch to you. Dear, where are we going? There we go. Okay, should we come across?
Okay, is that okay?
Sounds good.
Yeah. Cool. So why can't I see my screen? There we go. Is that okay? Yeah.
Looks good.
Brilliant. Cool. Sorry. Right. Thank you. Thank you. Right. Yes. I'm Emily, and I'm going to be talking today about my research on the role of the endoplasmic reticulum in plant immunity. Thanks everyone for coming along. Okay. So. Oh no. Sorry. Okay, so I just want to start with an introduction to the endoplasmic reticulum. The ER is the gateway to the plant secretory pathway, and as such, it's responsible for protein synthesis, folding, glycosylation, and quality control. The ER is also involved in lipid biosynthesis and is an important store of calcium in the cell. In cereal crops, the endoplasmic reticulum forms an important storage compartment for proteins and lipids. And through the unfolded protein response, the ER also is important in biotic and abiotic stresses. I just want to say that I'm not going to be talking about UPR or ER stress today, but it is obviously a big topic and very relevant. So the ER is a continuous and highly dynamic membrane network, which is composed of a combination of sheets, which are these large flat surfaces, which I'm hoping you can see with my mouse, and tubules, which are highly curved and joined together at these three-way junction regions. A quick note is that most, if not all, I think, of the images that I'm going to present today, the ER is labelled with the either GFP or RFP HDAL, and this is just simply the fluorescent protein tagged with the 4 amino acids HDEL, which acts as a ER retention motif and retains the fluorescent protein in the ER lumen, and is a really good way of lighting up the ER. Okay. So this image here is a 3D rendering of the endoplasmic reticulum in 2 adjacent cells, with the lower cell shown in red and the upper cell in blue. And what I hope this really demonstrates is just how pervasive the endoplasmic reticulum is, in that it extends throughout the cytoplasm and forms contact sites with most, if not all, of the other organelles in the cell. And consequently, the ER is likely to be integral to intracellular communication. Also, the ER extends between cells via the plasmodesmata, and therefore is also involved in intercellular communication. So you can see why it's quite an important organelle. Okay. So despite its complex shape, there is actually only a handful of proteins known to be involved in forming the ER morphology, and these fall into 3 main families. First of all, we have the reticulum family of proteins, which are involved in the formation and stabilization of ER tubules, and consequently they're mainly localized in tubules, but are also present on the curved edges of sheets. And this confocal image at the bottom just shows the sort of phenotype that we get in the ER when we overexpress a reticulum, and effectively we get over squeezing of the ER tubules, and consequently this then constricts the luminal contents into these discrete bulges, and you get this characteristic bead on a string type morphology of the tubular ER. The second major group of ER morphogens are the elastin family of proteins, which in plants are known as RHD3 and RAL1 and 2. And these are involved in the fusion of proteins together to form the characteristic network. And this image at the bottom shows the ER morphology when we overexpress a dominant negative form of RAL2, which is then limited in its ability to fuse membranes together, and so we get these long cable-like ER tubules. And the final group of proteins known to be involved in ER morphology are the lunar part proteins, of which there are 2 in Arabidopsis. And these are involved in the stabilization of 3-way junctions. And when we overexpress lunar park 1, we get a proliferation of, we get localization of lunar park at the 3-way junctions, and we get this proliferation of sheets at the junction regions. Okay, so a lot of my previous work was involved in investigating the mechanism of actions of both reticulums and Luna Park. And we published a number of papers on this. However, more recently, I've become more interested in how the ER morphology as a whole changes in response to different developmental and environmental cues. I said previously that the ER is a very dynamic membrane system. Okay. So a couple of years ago, we looked at how the ER morphology changed in Arabidopsis embryos during seed germination. And to do this, we decoated the embryos and then imaged the ER over 7 days. And so here you can see images of the ER and together with the corresponding appearance of the embryo at that time point. And what we basically saw that is from days 1 to 3, the ER didn't change much in shape. It had this quite tubular structure, but almost like a honeycomb-like appearance with these large circular spaces, and these tended to be filled up with lipid bodies in the developing embryo. However, around day 4 or 5, we saw a massive change in the ER morphology, and we saw a huge proliferation of ER sheets or ER cisternae. In fact, the ER was almost totally cisternal at this time point, and this corresponded with the embryo starting to green, presumably as the photosynthetic machinery is being synthesized. At day 6, we saw that the ER morphology was becoming more tubular again, and by day 7, the morphology of the ER resembled something that we typically see in a mature green leaf. Okay, so what this time series I hope quite clearly demonstrates is not only how plastic the ER morphology is, but also presumably how these changes in shape reflect the changing functions of the cells and of the whole organism. So because of this, I'm now really interested in what the fundamental relationship is between ER form and function. Okay. Okay, so my current work is actually looking at the role of the endoplasmic reticulum during plant immunity, and more specifically asking if we can manipulate the morphology of the plant ER to enhance the plant's defense response. And to do this, I'm taking a two-pronged approach. So I've got 2 major objectives. Firstly, we know from microarray and other transcriptional profiling experiments that there's huge transcriptional reprogramming during pathogen infection. And this isn't surprising because there's obviously a significant increase in demand for de novo lipid and proteins to be biosynthesized. And coupled with this increased demand for secretory proteins, this necessitates rapid ER expansion and remodeling. However, this expansion and remodeling cannot just proceed unchecked, otherwise you'll start to get accumulation of misfolded and unfolded proteins, which then triggers ER stress. So any ER expansion and remodeling must be highly regulated. Okay, so the first part of my project I was really interested in getting a handle on how exactly the ER architecture changes both spatially and temporally during the course of an infection. And then my second major objective is flipping this round and looking at it from the point of view of the pathogen. So if the ER is this majorly important secretory protein factory, it seems highly likely that it's a good target for pathogen manipulation as part of its virulence strategy via the secretion of small effector molecules. So with this in mind, I was interested in trying to identify whether indeed the ER was a core target for multiple pathogen effectors, and also whether I could identify the cellular target of these effectors to see if multiple effectors from different species converged on a common ER component, which would then suggest that that ER component was critical for the defense response. And ultimately, it would be really nice if we could then use these findings to re-engineer the ER at key stages in the infection process and thus positively alter the host pathogen outcomes. Okay, so the first part of my talk, I'm going to be concentrating on how the ER architecture changes during infection, and then I'll go on to talk about different ER targeted effectors from different pathogens. Okay. So when I decided that I wanted to look at how the ER was remodeled, I obviously needed to choose a pathosystem. And one that we've used extensively in the lab is the virulent pathogen, the virulent biotrophic bacteria, Pseudomonas syringae DC3000. And the advantages of this pathosystem are that, firstly, it's very easy to synchronize infections. And secondly, the whole infection process occurs over a matter of hours rather than days, and when it comes to imaging, that's a big advantage. So I can very easily image throughout the course of the whole infection. So to do this, I took Arabidopsis lines, stable Arabidopsis lines expressing RFPH style, which, if you remember, was the ER luminal marker. And I either did a mock infiltration where I infiltrated magnesium chloride, or I infiltrated with DC3000 stably expressing YFP. So in all the images that I'm going to present, the plant ER is in red and the DC3000 bacteria is in green. Okay, so this image here is an image of the mock challenge, and also it's pretty much what I saw up to around 7 hours post-infection. I should have said that I started imaging around 5 hours after infection. So up until around 7 hours, I don't really see a lot going on. The leaves in the challenge leaves look very similar to the ER in the challenge leaves, sorry, strongly resembles the ER in the mock control up to around 7 hours. However, after 7 hours, I really start to see changes in the ER morphology. So the first thing to say is that around 7 hours, the bacteria become really easy to spot. Before this time point, they were few and far between, but around 7 hours, they're pretty much everywhere. They're also highly mobile, and as you can see in this image, they are actively dividing. I also see an abundance of bacteria around the stomatal regions, but that's just an observation, really. So as I said, the bacteria around 7 hours are very mobile. Despite this, though, presumably even though they're not adhered to the cells, they presumably are still capable of activating the cell surface receptors. And this is an image not an image I took, but this is an image, an electron microscope image, which was also taken around 7 hours. And not only does it give you an idea of the size contrast between the bacteria and the plant cells, but it also shows the bacteria starting to embed in the crevices between adjacent cells. Okay, and this is what I start to see at between 7.5 and 8 hours is less mobile bacteria. By 8 hours, the bacterial colonies are now starting to really become established, and they are definitely localizing. They're definitely a lot less mobile, and they're starting to localize between adjacent cells, as you can see here, and this larger colony here. In terms of changes in the ER, well, I can't really see any obvious changes in the ER morphology by 7 hours, and arguably I can't really see anything definitive by 8 hours, although perhaps there are some early evidence of changes in the ER morphology. But by and large, the ER network strongly resembles what I see in the mock challenge. However, by 9 hours, I can clearly see differences in the ER morphology. The ER seems to form these, seems to condense into these nodular knot-like structures, such as I've circled here and here and here as well. The ER colonies themselves are now very noticeable and very large. And then between 10 and 12 hours, I start to see complete disintegration of the ER tubules, and this eventually leads to complete collapse of the ER network entirely by around 12 hours. Okay. So what I also observe is that this isn't a uniform response across all cells in the infected leaf. In fact, cells appear to be responding autonomously, and it's the cells that are directly adjacent to the bacterial colonies that I start to see these changes that I described in the previous slide. So if you look at this image here, you can see the bacterial colonies in green, and it's in these upper cells I start to see this nodular knot-like ER morphology, whereas this lower cell, which is more distal from the bacterial colonies, is still maintaining a lot of its network structure. And then secondly, and this is a bit more tentative, so so if we go back to my electron microscope image, we see that the bacteria are lodged between two cells, and presumably are equally capable of injecting their type 3 secretion system, which is used for the delivery of effectors, into either or both of these cells. However, on occasion, I do see discrepancies in the ER morphology, even between cells that are both adjacent to bacterial colonies. So this cell here, this is one continuous cell. The ER appears to be a lot more affected than this lower cell here, which is maintaining more of its network structure, despite both cells being adjacent to a bacterial colony. And this possibly suggests that there is an uneven distribution of effectors being delivered into the top cell compared to the bottom, but I'll reserve judgment on that. That's very tentative. Okay, so aside from DC3000 infection, I was also interested to see whether the ER morphology was similarly infected during PTI, so during PAMP triggered immunity. So to look at this, I infiltrated the leaves with known PAMP elicitors, including FLAG22 and chitin, and then looked across a range of time points, of which I'm presenting the images from 16 hours here. And in all cases, with my PTI elicitors, I didn't detect any obvious changes in the ER morphology in comparison to my mock infiltrated control. So these changes appear to be something that's exclusive to virulent pathogens that are capable of delivering effectors into the cells. Okay, so a couple of other points I'd like to make is that, so I don't see, just to summarise what I've just presented, I don't see a lot of changes in the ER up to around 7 hours, but then from 7 to 9 hours, I see some very rapid reorganisation of the cellular ER. And this time course very strongly resembles what we previously published about the implanter growth of DC3000. So in 2015, we published in Plant Cell and showed that there was no significant increase in bacterial numbers up until around 7 hours. And then by this time point, presumably the effectors have been delivered into the cells and are starting to suppress the host defenses. And then from 8 hours onwards, we see an exponential growth of bacteria. And these timings sort of almost directly mirror the timings that I see with changes in the ER morphology. The other thing I'd like to say is not only are these changes very early, but they also seem to precede the response of other organelles in the cell. For example, if we look at chlorophyll fluorescence measurements around a similar time point, so these are chlorophyll fluorescence images which indicate the FV/FM values of the chloroplasts, which is a measure of the efficiency of photosystem 2. In the first image, which was also taken at 9 hours, the two right-hand leaves were infiltrated with DC, and there's a mock infiltration here at the bottom, and we see very little change in the efficiency of photosystem 2 at this time point, and this is reflected in the graph here. And it's only around between 15 and 20 hours that we really see the chloroplasts function starting to be suppressed, as you can see in this second image taken at 20 hours. Whereas in comparison, the ER shows obvious changes in its morphology around a similar time. So these ER changes are, not only are they very rapid, but they're also early in the defense response. Okay, so I'm very aware that when I present all these images, I'm saying that, oh, the ER didn't look like it had changed, or this ER was very similar to that ER, but that's quite subjective, and it's certainly very qualitative. And this isn't really a robust system if we want to make, if we want to pick up early changes, or if we want to make direct comparisons between treatments or time points. So with that in mind, I said previously that the ER morphology in the leaf is very dynamic. So we can, although in a normal leaf, for want of better expression, we get a mixture of ER cisternae and tubules. This ER morphology can be changed to something that's highly tubular. For example, I showed at the beginning that if we overexpress the reticulums, which are involved in the formation of ER tubules, we get a very tubular network. And similarly, if we overexpress other things, such as when we apply the trunculin B, which depolarizes actin filaments, we also see a proliferation of ER cisternae. And this is, we also saw this, if you remember, when we looked at the embryos around day 4 and 5, the ER there was also highly cisternal. So a normal ER, in inverted commas, can be anywhere on this scale between something that's highly tubular to something that's highly cisternal. So because of this, we wanted a better way of parameterizing the images that we were taking. And to do this, we recently started collaborating with Mark Fricker at Oxford University, and Mark has developed this very sensitive and sophisticated algorithm called Analyzer ER, which performs quantitative analysis of plant ER architecture and dynamics. And this is really useful to enable us to not only compare between treatment and time points, but also to hopefully be able to pick up early changes in the ER morphology, which are statistically significant, but maybe perhaps aren't so obvious to the eye. Okay, so what Mark's algorithm does is it takes the image of the ER and it skeletonizes it and segments it into tubules and cisternae, and then we can perform any number of metric analyses on the different areas. So, for example, we can make measurements of the different tubule widths, which might be affected, for example, if there was an overexpression of reticulums. We can also look at tubule length. We can make measurements on the cisternae. We can do texture analysis, for example, because the cisternae aren't homogeneous. They actually show different light and dark regions. And we can also make measurements of cisternal area and see how these change between images. And finally, we can make measurements of the polygonal areas, which is the negative space, if you like, between the tubule network. And in addition to the static architectural image properties, we can also look at changes in the movement of the ER and its change in dynamics across different treatments. We've only recently started to work with Mark and to apply this algorithm, but it's already starting to prove really useful in being able to put metrics to some of the otherwise qualitative images. Okay, so to conclude that part of my talk, I hope that I've convinced you that there are, that the ER rapidly changes in response to pathogen infection, and that these changes occur early in the defence response. Also, that cells respond autonomously to the presence of pathogens, with those cells that are immediately adjacent to the pathogen seeming to change their ER morphology before more distal cells, and that the timing of these ER morphological changes seems to coincide well with the timing when we know that the cellular defences of the host have been suppressed. Okay, so with this in mind, it appears that a lot of the changes that I'm seeing in response to DC3000 infection presumably are either directly or indirectly caused by the injection of effectors into the cells. So I was obviously interested in identifying putative effectors that may be targeted to the ER. Okay, since it is likely to be a prime target for pathogen manipulation. Okay. So one of our collaborators in France, INRA, very kindly gave us a subset of effectors from the RXLR effectors from the sunflower downy mildew pathogen, which is an oomycete, Plasmapara halsteadii, and these effectors localized to the endoplasmic reticulum. And then a few years ago, Jonathan Jones's lab published a number of effectors from Peronospora, which also a subset of which also localized to the ER. So I got my hands on this subset of effectors and confirmed that I was happy that they localized to the ER in our hands, and then looked in more detail at the protein's structure. And when I did this, I saw that all of these ER localized effectors seem to share this similar protein topology, in that they had that the bulk of the protein was predicted to be localized to the cytosol, and then there was this C-terminal tail anchor transmembrane region with this small tail inside the ER lumen. And this perhaps wasn't a great surprise that they had this topology, as C-terminal tail anchors are a known targeting mechanism for post-translational insertion of proteins into different membranes, including the ER, but not exclusively the ER. It's also used to target proteins to mitochondria, for example. So we looked into this in a bit more detail, and last summer, I had a really good, really capable master's student called Vicky, and what she did is she looked at different truncations of one of these oomycete effectors, one of the sunflower downy mildew effectors. And what she did first was to clone the transmembrane domain on its own and fuse it to GFP. And then when she expressed this transiently in Benth, she saw that the oomycete effector was still capable of localizing to the ER. However, when she cloned the cytosolic portion of the protein without its transmembrane domain, it no longer showed ER localization, but instead was expressed diffusely throughout the cytoplasm. So as a result of this quite simple experiment, we concluded that this C-terminal tail anchor was both necessary and sufficient for the ER localization of this effector. Okay, so with that in mind, having proven that this was a robust way of targeting proteins to the ER membrane, we then wondered whether we could use this topology as a screening tool to identify novel ER localized effectors. So we decided to do this in a different oomycete, in Phytophthora infestans, which is not only is it economically important as the causal agent of potato blight, but it's also very closely related to Plasmapara halsteadii and also relatively closely related to Peronospora. So what we did is we created some quite stringent criteria in order to identify putative tail-anchored effectors that may target the ER in Phytophthora infestans. So first of all, we took the sequences of all 563 known RXLR Phytophthora infestans effectors, and we decided to define a ER transmembrane domain as being between 17 and 22 amino acids in length. And we didn't just pluck this figure out of the air. There was some logic behind it, because early papers on the reticulums and their mechanism of action identified that the width of the endoplasmic reticulum membrane was around 17 amino acids. So that's where that figure came from. We then also defined that for it to be a tail anchor, there needed to be a maximum of 30 amino acids downstream of the transmembrane domain. And this figure of 30 is an estimate of how many amino acids could be present in the ribosome exit tunnel after the hydrophobic transmembrane domain has been exposed. Okay, and finally, we really wanted to concentrate on effectors for which there was evidence that they were actually expressed during the course of an infection. So to do this, we mined publicly available RNA-seq data and concentrated our efforts on effectors for where there was strong evidence that they were actually expressed. Okay, so having applied these three different criteria, we then came up with a subset of 17 putative ER effectors which possessed this which met our criteria for a C-terminal tail anchor, and an additional 8 effectors which sort of nearly met it. Okay, so out of this 17 plus 8, we selected a subset of 14 for further analysis. We then cloned these 14 effectors and expressed them, created fusion proteins to GFP and transiently expressed them in Benth and looked at their localization. and we were reasonably successful. So we showed that of our 14 cloned RxLR effectors, 6 of them localized exclusively to the endoplasmic reticulum. These images just show the effector fused to GFP and co-expressed with the ER luminal marker RFPH DAL. And a further 4 of our effectors localized to both the ER and the Golgi. So these first 2 images are again co-expressed with the ER marker RFPH DAL, and these second 2 with the Golgi marker STRFP. In addition to these ER and Golgi localized effectors, we also found 3 that instead localized to the mitochondria, which perhaps wasn't surprising given as tail anchors are a known mechanism for targeting the mitochondria. I should say that 09218 has previously been published as going to the mitochondria, and it shows extensive similarity to 09216. So this was quite, as a strategy, this worked really well, and we've now got a set of ER targeted effectors, which we're currently working on identifying the host target in the plant. Okay, so before I wrap up, there's one other thing that I wanted to show you today, which was is a very novel result. We only got it just before we had to close the lab, so I just sort of wanted to put it out there really. So in addition to our tail anchored ER effectors, we also were given this other effector from this other RxLR effector from the sunflower downy mildew pathogen, which also localized quite nicely to the endoplasmic reticulum, didn't seem to affect the endoplasmic reticulum morphology in any way. However, unexpectedly, what I also saw when I expressed this effector in Benth was it seemed to the ER seemed to be intimately associated with the chloroplasts, and these chloroplasts seem to be clustering around the nucleus in the cell. And this perhaps isn't that unknown. Other people have published that that they've seen this reorganization of chloroplasts during infection with both virulent and avirulent bacteria, and it's believed that this perinuclear localization of chloroplasts may facilitate retrograde signaling between the chloroplast and the nucleus. This image here, I can't take credit for, but basically here it shows the chloroplasts in red and a second subset of chloroplasts which are stained in green, and these are stained with DCF, which stains for those chloroplasts which are actively producing reactive oxygen species. So you can see that the chloroplasts that are, and the nucleus itself, the chloroplasts that are clustered around the nucleus are actively having a ROS burst. And then this second chloroplast here, which has this stromiole, which is drawing it into the nucleus. So as I said, this perinuclear organization of chloroplast is something that's been seen previously. In addition to this clustering of chloroplasts around the nucleus, I also saw this this clumping together of chloroplasts wrapped in the endoplasmic reticulum throughout the cell. And one final image is, at first glance, this looks like stromioles emulating from chloroplasts, but actually, because we know that this is ER that's being labelled with GFP, this is actually ER network and ER tubules, and because I also see this polygonal structure here, I am quite confident that this is ER that I'm actually seeing. But this is quite reminiscent of the stromioles, and it almost appears as if the ER might be drawing the chloroplasts into the nucleus. So this is all very tentative at the moment. I just wanted to present the images. I'm not making any outright statements about what's going on. I'm obviously keen to investigate this further, or if anyone else has seen anything similar with clumping of ER and endoplasmic reticulum, or wrapping of the endoplasmic reticulum around chloroplasts around the nucleus, I'd be interested to hear about it. Okay, so just to draw to an end, in conclusion, I'd like to say, I really hope that I've convinced you today that the ER is critical to the perception and regulation of adaptive host responses to biotic stress, and that it rapidly remodels upon pathogen infection. One thing I said right at the start of my talk was that I was interested in the fundamental relationship between ER form and function. So I've done a good job today, I hope, of describing how the ER changes shape, but I haven't really touched upon about whether these changes in the ER morphology have any impact on the susceptibility of the plant to pathogens. So to try and address this, I have some preliminary data. Oops. So here we have, this is an Arabidopsis plant stably expressing GFPH style. So this is a sort of pseudo wild type ER morphology. And then this Arabidopsis line is overexpressing the chaperone calnexin. And when we overexpress calnexin, we see that the ER mainly exists in cisternal form. And then what we did with these two lines was we drop inoculated them with botrytis spores. Botrytis is a necrotrophic fungus, and then measured the development of lesions over the next 72 hours. And what we quite clearly saw is after 48 hours, but even more so after 72 hours, there was a significant decrease in lesion area in the calnexin mutant. So although this is preliminary data, it does suggest that perhaps an ER that is more cisternal in nature then renders the plant more resistant, at least to necrotrophic pathogen infection. And I need to explore this further and see if it extends to other altered ER morphologies. But it's encouraging, at least, that suggesting there is a link between form and function. Okay, and secondly, I hope I've shown that the ER is indeed a target for multiple pathogen effectors, and that a subset of these are targeted to the ER by virtue of possessing this C-terminal tail anchor, and that we can use this topology as a way of screening and identifying new ER localized effector proteins. Okay, I'd just like to thank everybody at Warwick, and particularly my PIs, Lorenzo and Murray, Mark at Oxford, Hazel and Petra at the James Hutton Institute in Dundee, because they helped me with a lot of the Phytophthora work, and Catherine and Sarah at York, who did the, well, Sarah did the botrytis assays for me, and also Laurence Godiard at INRA, who donated the sunflower pathogen effectors. And one final thing I'd like to just shoehorn in is that if anyone knows any capable and willing students, we have actually got a funded PhD studentship on offer for 2020, investigating the role of the ER in plant immunity. And that is it. Thank you, everybody.
Thank you. Thanks very much, Emily. That's great. And I think that's really great that most of that stuff you showed was completely unpublished, right? So it's really, really cool new stuff. And that's, yeah. You know, we want to encourage that for people. I'm not getting at you there. I'm just saying it's cool.
No, it is. I'm in the process of writing it up and pulling it all together. Yeah.
Excellent. That's cool. So we had a number of questions come through, and so it's great that you people were asking us at the start about fungal pathogens, but you touched on bacteria and oomycetes and fungus as well. So that's great. So first question from William Rooney, and he asks, does the expression of the effectors that you use, does that result in any changes in ER morphology at all?
No, no. In their own right, I don't see any obvious changes in ER morphology as a result of expressing the effectors. That said, I've expressed them transiently in Benth, and I don't really see anything, and that's 3 days after infiltration. Although I know that the people in Petra and Hazel up in Dundee have said that they do see changes in the ER morphology when they express different effectors, but that's not something I've particularly seen. And also, when I've made stable Arabidopsis lines of these effectors, I don't see any obvious change in the ER. But I would caveat that by saying that when we also don't see changes in stable lines when we alter the expression of reticulins either. It's almost as if the plants adapt to having altered reticulin expression and adapt their ER accordingly, so that the same thing might be going on when we try and make stable lines of the effectors.
Okay, that's cool. So a lot of there's a few other questions along those lines. Actually, there's quite a lot of interest in the effectors and how they're interacting. So Katja Hugard from asks, how do you have any idea, or either you or your collaborators have done, do you have any effect, any idea how the effectors interact with the ER at all? And any idea what they're doing there? Any sense of that, or is it too early to say at this stage?
No, it's not too early to say. So there's not much has been, well, not much has been published on the ER in defence anyway, but there are some papers out there, and the group at Paul Birch's group at Dundee have published on one ER localised effector from Phytophthora infestans, and this is known to target NAC transcription factors that are localised on the ER membrane. So there's a subset of NAC transcription factors that actually have a transmembrane domain and are under uninfected conditions. They are localized to the ER, but then something during an infection or during other stresses, something then triggers that transmembrane domain to be cleaved and drives the and then the rest of the NAC transcription factor then moves to the nucleus. And what Paul's group has published is that one of these oomycete ER targeted effectors prevents effectively locks the NAC transcription factor on the ER and prevents its relocalization to the nucleus. And I've done yeast to hybrid experiments on all these membrane locked, they're called NTLs, this subset of NAC transcription factors. I've done yeast to hybrid on my ER targeted Phytophthora effectors, and a number of them do interact with these NAC transcription factors. And also there's a paper published last year which similarly identified effectors from Bremia, the lettuce oomycete, which also seem to target these NAC transcription factors. So these do seem to be a common target, but presumably there are others as well.
Okay, cool. So we got a question from Guido van der Akkerwecken, who asks, do you have any idea how those proteins travel from the pathogen into the plant cell, since they've got the membrane anchor, and how might you test that?
Oh, that's a good question. Well, obviously, when I've expressed them, I've infiltrated them, agro-infiltrated them directly into the leaf. So that sort of circumvents that. What would be really nice is if I could get them to be delivered via the oomycete, and then we could really get a handle on what they're actually doing in a perhaps more realistic infection process, but that's not available at the moment. Okay. No, to be honest, I don't. I could only speculate about that.
Okay. All right. So a question from Stefan Wojcik. He asks, was there anything specific about those tail anchored proteins that determined their localization? Do you know anything about the sequence or the structure of them?
Yeah, I do. So I tried to look in quite a lot of detail about whether there was anything specific about the transmembrane domains. So in terms of the length of the transmembrane domain, there didn't seem to be anything specific that would determine whether it went to the ER or the ER Golgi or the mitochondria. One thing that I did find is that the hydrophobicity of that transmembrane region did seem to be a lot higher in the effectors that went to the ER compared to the 3 that went to the mitochondria. But you've got to remember, I've only got 3 mitochondrial effectors, and 2 of them are very similar in sequence. So I wouldn't want to hang my hat and say that the hydrophobicity of mitochondrial transmembrane domains was lower, but at the moment it does look that way. But that's the only physiochemical property that I could obviously identify that distinguish between ER and mitochondrial tail anchors.
Okay, cool. Okay, so a question from Cyril Zipfel, and he asks, I don't think he missed this. So obviously at the start you showed that the Pseudomonas caused some morphological changes in the ER. And do you see any morphological changes with the oomycete or the fungal pathogens at all?
No, I don't. I don't, if I'm honest. And the timing would be harder to look at. The benefit of looking at the bacteria is that it's all done and dusted from between 3 and 18 hours, so it's quite easy to image. But with the oomycete, it would mean imaging, doing time-lapse imaging over a number of days. That's not saying that that's not worth doing, and it certainly would be interesting, but I haven't really got a handle on what the most informative time points would be during the infection process with Phytophthora, but that is something that I'd be keen to do, yes.
Okay, so got some more general questions, maybe one from Matthew Terry. So he asks, and this is again, and there's also a question about from Stuart Sullivan about light and about ER morphology in general. So, okay. But the question from Matthew specifically about whether the ER morphology, does it come from the plant? Does it change it, or does the bacteria elicit or cause the change in the morphology? Can you distinguish that between those two things?
That's what I'd be really interested in finding out. I mean, obviously, with any of when you've got two living organisms in a system, it's hard to know what's cause and what's effect. The big gap, which I'm surprised nobody's picked up on in my results, is that I really need to look at the ER morphology in the presence of bacteria where the type 3 secretion system's been disabled, and that's what I was about to do when we had to shut the lab up. So in that will at least answer the question whether it's merely the presence of the bacteria in the cell that's causing these changes in the absence of effectors, or whether it really is the effectors that are causing it. I've got some preliminary data from E. coli, from a fluorescent E. coli, which seems to suggest that the presence of E. coli doesn't noticeably affect the ER morphology, but I know I really need to do it with the heart mutant, and I think that will go a long way to at least trying to unravel what exactly is causing these changes. And likely it's a combination of both. It's a combination of the ER increasing its the plant increasing its production of antimicrobial secreted proteins, and on the flip side, the pathogen trying to manipulate this factory and cause UPR stress and shut down this increased protein production.
Okay, yeah. So there's a similar question from Ho Hang Go, who asked whether it's causal or a consequence of the pathogen invasion. I think you answered that there. So yeah. Let me ask. So question from Peter Wally. So he asks about other tissues. So you look at leaves here exclusively in what you said here. So has anyone else looked, or have you looked at other tissues to look if there's any effect on the ER in this case?
I haven't. I've only looked at leaves, and that's all I can say on that.
Okay.
No one else has looked. As far as I'm aware, it's quite a neglected area. Sorry, excuse me. Not many people have. Well, I haven't seen any evidence of anyone else looking at of really doing in-depth studies of how the ER in other tissue changes now.
I think probably partially because it's incredibly difficult to quantify, but it looks like you're starting to get your head around that a little bit with your collaboration.
Sorry, I perhaps should have said this earlier. There are studies out there, some from the mid-90s, which showed that the ER, sorry, that the ER does remodel around historia, and that the historia might be, in fact, the source membrane of the extrahistorial membrane. So there are papers out there that show that the ER does remodel in the immediate vicinity of the historia.
Okay. So a question from Yasin Dagdas, and this is about some of the stuff you start at the very start. So the calnexin overexpression, there's some changes in ER morphology, and do you have any idea why that happens?
No. I mean, it would be really nice to be able to say that ER sheets are associated with ribosomes. Ribosomes are associated with protein production. So the more cisternae you have, the more protein production that you have. That would be a really nice conclusion. But I haven't got conclusive evidence that that's the case, and that's not been published either.
So a lot of people actually, a lot of people are asking a lot more general things about, have you done this? Have you got evidence of changes with symbiotic bacteria? Have you looked at other tissues, things like this? And as you just said, this is a very ill researched area. So you're really leading here. So there's not a lot of extra stuff in that. So let me finish with a general question from Jocelyn Benitez Alfonso, who talked last week, 2 weeks ago. So, and about the tools that you have in the ER. So she asked if there's a marker that you can distinguish between different parts of the ER. So desmotubule ER at the plasmodesmata, and just ER in the rest of the cell. What sort of markers have you got that allow you to distinguish between ER in different places?
We haven't got any markers that distinguish. It would be really nice to there is some published evidence saying that there are sorry, there are different subdomains of the ER, and it would be really nice to be able to distinguish between those and see if we're getting an enrichment of certain subdomains over others. But in terms of markers for different subdomains, I'm not aware of any that are being routinely used. In terms of desmotubules, one of our collaborators at Oxford Brookes published a few years ago that reticulum 3 and 6 are only found in the plasmodesmata, so presumably they could be used as markers for the for plasma alongside other plasmodesmata markers, but they could be used to identify the desmotubules that go through the plasmodesmata. But that is all I'm aware of off the top of my head.
Okay, all right. So I think we'll leave it there. Your voice is going, but so, thanks very much, Emily. So the timing, you perfectly expanded your talk to fit into the hour. We'll give you the benefit of the doubt for that. So it's been really great to hear this work. And sorry to a few people who we didn't get to their questions. And I'd encourage you to sign up for the talks in 2 weeks' time and in 4 weeks' time, and there will be announcements coming soon about future talks as well. And write to me if you're interested in